there are several maladies, several diseases for which we do not have optimum therapies. We today have moved from chemical small molecule space to recombinant products, monoclonal antibodies. However, there are several disorders for which we do not have a therapy, or we can say nil therapy. So the first topic that I want to talk to you is on the unmet medical needs in the country, including not just the country but also pan world or global scenario. It is said that there are several rare diseases identified globally for which there are single point mutations or single gene defects, and about half of them are rare diseases which affect the child population. Looking into these issues, the government of India has developed a policy which is called the National Policy for Rare Diseases. It was enunciated in 2021, and this document says that it is very tough and to identify these diseases in the vast Indian population, but if you first identify, diagnose, then we can start developing various products. It has reported that there are several lysosomal storage disorders, inborn metabolic errors, and including spinal muscular dystrophy. These are inborn defects which are very difficult to treat with small molecules, and there is no other therapies except for the gene therapy and targeted drug delivery systems for these type of genetic disorders. Most of these diseases are life-threatening, and you will not find the child having proper growth, or they do not even come up to a level of self-dependency. They will have to depend upon several other health caregivers, including their families. And it is heart-touching to see this population for which we are unable to provide any therapies or rescue. Apart from these genetic disorders, we also have the neurological disorders like Parkinson's, endocrinological disorders like diabetes, type 1, type 2, and various other cancers where gene and cell therapies can provide a solution. So I also want to touch upon the flagship program that I was just saying. The government of India has identified the National Sickle Cell Anemia Elimination Program in its union budget in 2023. It addresses on identifying patients with sickle cell disease and aims to eliminate the cell genetic transmission by the year 2047. And this is a mighty big program, considering that sickle cell anemia is devastating disease and there is no other therapy except for blood transfusions. You must be thinking, so what are the other therapies that the government of India wants for these type of patients? CSIR, a government of India laboratory, has generated a CRISPR protein, and they are developing the product further to do the proof of concept and then proceed to the clinical trials. This is the most important achievement and a milestone for India. Have you all ever heard of this program, National Sickle Cell Disease Program, and the protein replacement through targeted gene and gene editing therapy is next big history that India wants to create. Recently, US FDA has approved 2 products which are Casgevy and Lyfgenia. They act by genome editing technology, and Lyfgenia is a cell-based gene therapy. So it's not all that dark, and India has progressed in recombinant products as well as gene technologies. So coming to the Drugs and Cosmetics Act and the various regulations today, I have shown you in this slide the step-up approach. Initially, we started with the new drugs and clinical trial rules in 2019, which was a substitute, which is today the substitute for the Schedule Y. Schedule Y is applicable for veterinary products, whereas the NDCT is for the new drug development and clinical trials. Considering that there are several cellular therapies available today, the Government of India in 2021 further clarified by providing what is regulated by the CDSCO as cell therapies and what is to be regulated by the medical professionals themselves as a therapy. Subsequently, as a third step, we have brought in further amendments which I'll discuss and touch upon, which talks of organ on a chip and other simulated models which can be used to substitute animal studies. This is the Section 33P guidance issued by the Government of India on 9/2/21. But prior to this, I should touch upon the new drugs and clinical trial rules. Hitherto, there was no definition of cell, stem cell, or gene therapeutic products, or including xenografts, and specifically biologics or similar biologics. These definitions were brought in as new drugs. In our country, a new drug means for the first 4 years, it continues to be a new drug, and CDSCO has to approve this product, that is the licensing authority DCGI. However, in the case of vaccines, recombinant products, gene therapeutic products, these are perennially new, meaning that any manufacturer who in India, indigenously or a registered importer wants to bring these products from the international market, they will have to submit their dossier to CDSCO, meaning this is continuously a new drug and it is beyond the 4-year period. Today, we see there are several cell and gene cellular products that are being used as therapies, say for osteoarthritis, and the control on these products is not with CDSCO. They are typically managed by the medical professionals through their code of conduct, and it is similar to procedures like the bone marrow transfusion, etc. However, to bring in regulation and to define what a product is and to say what is manufacturing, we have brought in the Section 33P of the Drugs and Cosmetics Act guidance or direction, I should say, which says that if there is a product developed through substantial or more than minimal manipulation, through a manufacturing process of enzymatic treatment, altering the cell structure, cell biology, and its functions by minimal or manipulation, here you can think manipulation step is a manufacturing step, in fact, an ex vivo manufacturing or maybe even in vivo manufacturing or administration of a gene therapeutic product. So wherever there is manufacturing which would alter the cell biology and its biodistribution in the body has been brought under the control of CDSCO, and these products will be governed under the Drugs and Cosmetics Act. So the new drugs and clinical trial rules has various schedules. It's a battery of procedures that are very well laid out and defined. It talks of the general principles and practices for clinical trials. It also talks about the various procedures for approval of new drugs in the country for manufacturing indigenously or for import. And I would want to touch upon Schedule 2, which is basically for the non-clinical studies. We are not calling it as preclinical studies anymore. We are saying these are non-clinical studies. India has also a unique compensation structure under the Drugs and Cosmetics Act and DCT rules. This defines a very structured formula for compensation for serious adverse events caused in clinical trial. We also have a very structured schedule for the post-market surveillance under the new drugs and clinical trial rules. So reading the new drugs and clinical trial rules from definition to W and the Section 33P guidance issued by the Ministry of Health, we can come to the conclusion as to what is a cell and stem cell derived product, a gene therapeutic product, xenografts, etc. I will read it out here. The cell and stem cell derived product means a drug which has been derived from processed cell, stem cells, and which has been processed by means of substantial or more than minimal manipulation, with the objective of propagation and or differentiation of a cell or tissue, cell activation and production of a cell line, which includes pharmaceutical, chemical, enzymatic treatment, altering the biologic characteristics, combining with non-cellular components manipulation by genetic engineering, gene editing, and gene modification. It also talks of gene therapeutic products as a biologic that contains genetic material which are introduced into the human body for the purpose of treatment of disease, etc. This may also include nucleic acids, GMOs such as viruses, bacteria, fungi, engineered site-specific nucleases used for human genome editing, and ex vivo genetically modified human cells. This is not very well laid out, but I have put this as a slide so that we understand the concept of what is cell and stem cell derived product, and also gene editing and genetic engineered products. So how different are these products from the clinical pathway, or what is the roadmap for these clinical development at ATMPs, cell, stem cell, and gene therapeutic products? Today, it is not very well defined under the NDCT rules. The schedules and the NDCT per se talks about conducting of clinical trials for new drugs, and which is more oriented to the small molecules or the chemical space and not specifically for biologics. For MABs, monoclonal antibodies, recombinant products, we have the biosimilar guidelines. For cell and gene therapeutic products, we still do not have any specific guidelines from the regulatory authority, but there are certain national guidelines published by ICMR, DBT, and ICMR. So briefly, I captured the essence of the issues and what will be the roadmap for clinical development of these cell and gene therapeutic products, which are so much necessary and essential today to meet the unmet needs. First and foremost, the difficulties arise as to how to demonstrate a human cell biology-based product, and how do we demonstrate their safety or pharmacology, toxicology in small animals. So the animal models is a big challenge here. Whether we need to look for larger animal models or mammalian models where we can prototype the mechanism of action, understand the mechanism of action of these living cells or modified cells, which are nothing but live drugs, is one of the challenges in animal toxicology for these type of products. However, one good thing that we brought in under the NDCT is the flexibility in the preclinical studies or the non-clinical studies. We have said that you can follow the ICH non-clinical methods, and also with a view to reduce, refine, or replace the number of animals that you use in your experimentation. If you look at the non-clinical studies and the characterization for pharmacology and toxicology that is wanting for these products, you will see it is a tough job to demonstrate their safety and efficacy prior to dosing into humans. The cell and stem cell derived products, first and foremost, must demonstrate the mechanism of action, the local and systemic toxicity, most importantly, the tumor-inducing potential and the ectopic tissue formation, their survival in the human body, persistence, their phenotyping and genotyping. More importantly, here we call this as, in place of ADME, we call it as biodistribution. So the challenges with cell and stem cell based products is rapid proliferation, and then creating additional ectopic tissues, which can also be cancerous. In coming to the gene therapeutic products, the most important tests that have to be done in terms of safety and safety, pharmacology and toxicology, is looking for the mode of delivery, that is whether we use the viral vectors, and if we are using viral vectors, is there any chance for reactivation, and whether these vector proteins that has developed during the production can cause viral replication or resurgence or reactivation. Whether the gene that we are targeting will be site-specific, and whether there will be any insertional mutagenesis, oncogenicity, vector biodistribution and transgene expression levels. These are the important factors that have to be identified, controlled during the preclinical or non-toxicology, non-animal testing, both put together to identify the dosing and also the reduced toxicity. Apart from these structured non-clinical studies and proving the safety of these products, understanding the mechanism of action, like any drug, they have to undergo the phase 1, 2, 3, and also the phase 4 trials. However, considering the unmet need and the requirement for these products, we can have adaptive designs, typically the phase 1 by 2 studies, or a small phase 1 study and then a phase 2 and product approval, followed by very structured post-marketing surveillance, because we are in need of these products. A recent change to the NDCT has been GSR 175E. Under this notification, we have brought in other non-clinical testing methods, that is cell-based assays, organ chip and microphysiological systems, sophisticated computer modeling, other human biology-based test methods, including animal studies, under the NDCT scope. This means today, people who want to take waiver from the non-clinical studies can use these systems and methodologies to identify the safe dose and also proceed for the human studies. So, having understood the complexity of these products and also the chances of ectopic tissue formation, oncogenicity, and understanding the safety of these products in vitro and through animal models, I want to focus on what are the methods and modalities that we have brought in the NDCT to enhance product development in the country and to provide drugs on a fast-track basis under the unmet need category. We have identified the international regulatory scenario which can boost and when implemented the same aspects in India. We have brought in a rule called the orphan drug for orphan drugs. We have defined the population. If the product is meant for less than 5 lakh population in India, then it will be treated as orphan drug. And what are the advantages that a manufacturer would get is there is no fee for conducting of clinical trial. The process for approvals can be accelerated and expedited. And this is a pathway which the cell and gene therapeutic products can actually uptake. On par with the US FDA, we too have brought in the accelerated approval processes in very special situations under the NDCT. The accelerated approval process may be allowed for a new drug for serious life-threatening diseases, rare diseases, diseases of special relevance to Indian health scenario, and for unmet medical needs, disaster, or special defense use as well. However, there should be a prima facie case of the product being of meaningful therapeutic benefit over the existing treatments if you want to use the accelerated approval pathway. Apart from the accelerated approval pathway, we also have the expeditious review process. Sorry, one more slide skipped. Yeah. So, what are the key features of the accelerated approval process and how do we do it? Typically at phase 1 studies, we try to understand the safe dose, whereas at phase 2, we are in a small set of population, we are trying to identify the dose, but phase 3 would be required in most of the cases for the small molecules. However, in unmet need conditions, particularly in the space of oncology, a surrogate endpoint such as 90-day overall survival instead of long-term survival can also be used. So we have set developed products. The timeline for review of the product dossier is 30 working days. It's just 30 working days. And if you don't hear from us, it will be deemed approval and the applicant can file a CT4A and proceed with the clinical trial protocol for investigational new drugs developed in India. For other applications, it is typically the protocol vetting time is 90 working days. And beyond the 90 working days, if you don't hear from CDSO, you can notify us and proceed with the trial. These are the new key features under the NDCT clinical trial rules. After undertaking the clinical trials, the next steps would be the marketing authorization. Under the NDCT, we have brought in CT23, which is a form for marketing authorization. And subsequently, the manufacturing licenses will be issued by the state licensing authorities. Another key feature of the NDCT is to promote research by MSME and also academic research. The fee for MSME has been reduced by 50% of the specified fee of the NDCT. For government and autonomous institutions, no fee for application to conduct CT. Autologous products which you need to track and trace even during your marketing phase and during clinical trial as well. Apart from the CMC manufacturing process validations, the non-clinical data has to be submitted. Then you proceed with the clinical trial protocols for phase 1, therapeutic exploratory trials and therapeutic confirmatory trials. The most important is the risk management plan and pharmacovigilance for cell and gene therapeutic products, because these are living drugs, living cells, or they retain in the body for a lifetime or maybe beyond that. Therefore, the risk management plan, identifying the patients who have been given these gene therapeutic products and monitoring them for adverse events or also even for the efficacy endpoints, is very important. Here I would like to present the recent approvals given by CDSU in the past few years and also the upcoming pipeline developments. CDSU has approved a few products which are cell-based as well as tissue-based. The first trial talks of human bone marrow-derived cultured pooled allogenic mesenchymal stromal cells, and also other products from the mesenchymal stromal cells, which are not stem cells per se, but these are ex vivo culture. We have also approved autologous adult live cultured chondrocytes for osteoarthritis, dendritic cell immunotherapy product for cancer, adult live cultured osteoblasts again for osteoarthritis, adult live cultured buccal epithelial cells for urethral strictures, and bioengineered composite limbal epithelial graft for keratopathies. And the recent approvals that were given was that of CAR T cells for malignancies, hematological malignancies. Unfortunately, we do not have any CAR T cells for tumors, but it's basically for hematological malignancies, B-cell lymphomas, and multiple myeloma. After 2023, the recent approvals, particularly for the humanized CAR T of IIT Mumbai, and subsequently to another manufacturer who brought in this product from Spain, we see at CDSU a lot of interest that has been generated in the development of CAR T cells, again for hematological malignancies. These are typically anti-CD19 or a combination of anti-CD19 and 20, BCMA antigens for multiple myeloma, and the newest of the block is anti-Claudin 18.2 CAR T cell for the indication of solid tumors. Solid tumors are really tough to handle through CAR T cells, and here we have a company who is coming up and is in the early development phase for solid tumors. Here in Hyderabad itself, we have big institutes who are doing trials and also develop products for retinopathy, and these are induced pluripotent stem cells, human derived, and meant for retinal pigment epithelial cells for the treatment of geographic atrophy secondary to dry age related macular degeneration. I'm nearing the conclusion of my presentation. And the most important and the flagship program is, like I said, the sickle cell anemia mission, where Government of India, through its CSIR labs, has developed a protein which will be used for gene editing for treatment of sickle cell anemia, and it is progressing very quickly. The proof of concept studies have been completed and they will be progressing slowly for clinical trials. So you must have noticed, all in all, that India is not far behind. We are picking up in the space of particularly oncology. However, there's a lot of unmet needs still in metabolic disorders, and we do not have a curative therapeutic agent from the cell and gene therapeutic space, at least for lysosomal disorders as of now. But we hope that the new drug and clinical trial rules will be further strengthened to give a clear regulatory pathway with the experiences and the lessons that we have learned during the approval of the recent CAR T cells, which is a milestone achievement for the Government of India and also IIT Mumbai. A good round of applause is really wanting for IIT Mumbai for the first CAR T cell and the humanized CAR T cell in India. I think if you are Mumbaikars, you'll be really proud of it. Of course, as India, we are very proud of this humanized CAR T cell developed by Dr. Rahul Purwar, and hope with this we will have good therapies coming up in not just hematological malignancies but also for solid tumors and other lysosomal disorders. Thank you for your attention. CRS. Successfully, with the availability of tocilizumab today, the CRS can be reduced, controlled, and therefore we should hopefully ensure that these patients survive long. Internationally, when given in childhood stage, the patients survive beyond 15 to 20 years. So if that was early identified, I think it's a very good success rate and a great hope for hematological malignancies.
Any other questions from the audience?
If you do not have questions, I'll answer you further. So it is another product that will be coming into the market shortly from Novartis. It is for SMA gene substitution and gene therapeutic product that sells at perhaps 17 crores. In India, they are ready to provide at 8 crores. In low and middle income countries, it is around 5 crores. So this is another gene therapeutic product. But if India has a skill set, the know-how, the research and technologies, I think we need it to manufacture these products, bring in these technologies, manufacture in India for our patients.
Thank you. Mike may not come there. Madam, you can come here. Come on.
Ma'am, I want to know that, like you said, ma'am, more and more people are coming now with these products at a lesser cost. So I think these products must be patented. So how
important is the vector and the transgene. The transgene, which is recombinant product, and vector is a delivery mechanism to alter the T lymphocytes. When the transduced vector is, used ex vivo to alter the cell, T lymphocytes, which are the fighting cells, they develop some receptors. These receptors would then lead it to the tumor or the malignancy and kill the cell tumor at the site itself. These are very targeted therapies. So to make a product where you develop a receptor, what's the key here is the plasmid or the transgene. So manufacturing the transgene is the key here. I also understand the vector system, lentivirus vector systems are also being brought into the country by import. However, Government of India has hit upon a very big project and we will be manufacturing LVV in the country today and not far away from Delhi. In a huge hospital center, a huge manufacturing site for lentivirus vector. Apart from the vector, if you have a technology that is the plasmid, which is very specific to the malignancy, then you can start, doing at the bedside of the patient, the manufacturing technologies. There are other companies called Miltenyi Biotech. I'm not propagating anybody's specific company, but these companies have come up with desktop manufacturing. Take the patient cells in a bag, T lymphocytes, manufacture it in this tabletop manufacturing equipment. The output is tested for sterility, for viral markers, cell genotype, phenotype, maybe not phenotypes particularly at the hospital center or the manufacturing center and retransfuse back to the patient. So what happens with these decentralized manufacturing is it's like apheresis machines in every hospital. Beside the patient, you can keep this machine and start manufacturing the same platform technology. If it was an Indian researcher or scientist who can share these technologies, and I'm sure they are ready to do that in the interest of patients in India, particularly because it is coming from IIT Mumbai, and these are all government organizations. We also have other companies, private companies in this space, which are coming up with various transgenes to target these diseases. However, the success rate, as one of my colleagues asked, is not very well known as of today, maybe over a period of time. Therefore, when CDSU approved these products, we said the condition is you monitor these patients for a period of 15 years. 15 years. It's not just 5 years or 3 years or 4 years, but they have to be followed up for long-term effects, including malignancy, secondary malignancies. Leave alone curing the primary malignancy, but there can occur secondary malignancies. So monitoring these itself is again on the manufacturer. These are expensive as such for fabrication, but also to get the safety data through long drawn trials. So these are going to be expensive. And if we have more technologies, bedside technologies, instead of shipping the product from one place to another, some of them have half-lives which the stability is in few hours. So you can't really ship the machine to the patient bedside. And that is where really manufacturing technologies or manufacturers or medical device manufacturers, should come in handy. There is a huge requirement for these type of technologies, and we can actually tweak the transgene to suit the specific malignancy. So that's the advantage of the CAR T cells. Here, there are so many methods to deliver the gene of interest. Ultimately, it should be specific, and the long-term effects of these products is very, very important, right? Lipocomplexes, there are so many, the major challenge is not just the gene construct here, is delivering it at the particular site and ensuring that there is no other insertional mutagenesis, etc. And these are drugs, like I said, these are living cells, living drugs, therefore could have off-site target effects, which are another issue. So safety is also the major issue in these products, apart from cost, the technology, manufacturing, etc. But we definitely need more specific technologies.